For example, if I take this cup of coffee that I drink, even the way the tip is designed as a function, so that the coffee doesn't spill over. The handle is a function to the cup. In a very similar manner, actually, one can draw parallels to structure and function. The only thing is, what is the function is what changes. So, in the sense that when we talk of biology, your function is the function of the protein,
Right.
or function of a machine inside a cell.
Ladies and gentlemen, thank you so much for joining us again on Ask How About. I'm your host, Pranay J. Reddy, and goddamn, it's good to be back. It really is. I'm so happy that I'm back with you all, staring into the void of this blank screen. Now, we've been away for over a month, and in the past month, a lot has happened. Not just in terms of the vaccine and the coronavirus, but also in terms of something really weird, the monolith. Now, I don't know how many of you have been following the story, but it's kind of weird to think that they all of a sudden started to appear randomly, and nobody knows who placed them or how they came to be in the Utah desert, and then they started appearing in Europe as well. Now, the going theory is aliens came and placed them here. And to the aliens, I ask, what do we Indians need to do to deserve your attention? I mean, seriously, even aliens, just like the Hollywood movies, aren't considering us to be a threat. Not even one monolith is present in India. Now, after this monologue, if there is a monolith present in India, God help us all. Now, coming to the good news, or rather great news, the vaccine is being administered to the general public in the UK. And as you can see here, the first woman getting administered is highly enthusiastic. Now, granted, she's 90 years old. She might be enthusiastic, we just might not know. But it is great to know that one person officially can roam around free in this world. And just like every 90-year-old, she can... Wait, don't 90-year-olds just sit at home anyway? No, on a serious note, I am glad that she's vaccinated. That means she can roam around free. And the second person to get vaccinated, and I kid you not, is named William Shakespeare. God damn, this vaccine's got superpowers. It's bringing back people from the dead. Now I know what you're thinking. Now we can finally have the story of Romeo and Juliet in the modern version, where all conversations happen through text. Hey, I think I kind of love you. OMG. Really? I think we should get, totally married and all. Nuh-uh. I'd rather be in a tomb and die. On a serious note though, seriously, I'm so glad this vaccine is here. About damn time, really. And I really hope it gets to other countries, and especially to India, as fast as possible. And I really hope it works everywhere and everyone becomes safe as soon as possible. Now, on the scientific aspect, this is an mRNA type vaccine. Now, what is that? Scientists take a part of this viral RNA and then insert it into the body. Now, unlike the full viral RNA, this RNA does not have any virility. So it cannot infect a person, but rather it only produces part of the virus which the body can recognize as foreign and then attack it. So when the virus comes to the body be like, what you doing here, son? Now, we have attached a research paper detailing how this vaccine works. In the link below, so do go and read that. It's very informative. Now, how do we know what part of the RNA, what to attack and what not to attack? So, in the words of our guest today, Dr. Balaji, every function of anything biological or in life has a certain structure to it. Now, unless we understand that structure, we won't know what the function is. So, it becomes highly critical for all of us to understand the structural part of anything if we want to understand it better. So that is why I personally believe structural biology is a very important field, and it will become, or rather take much more prominence in the coming years. So let's understand what it is and what goes on in it, all the tools, the techniques, the interdisciplinarity of structural biology with the interview with Dr. Balaji. Dr. Balaji, thank you so much for joining us today. I'm so glad you could be with us today. And how are you, sir? How's the pandemic been for you, or rather this, year 2020?
Yeah, it's been good and bad, both ways. Bad because I don't get to meet people physically, and there is nothing like real life interaction. Good because you can be lazy and put on a few kilos.
I completely agree with you there. It's like, yeah, I can get up in the morning in my bed and just attend class. I can't do that if it weren't for
Yes.
that. But
Then
yeah,
you cut down traveling time, you cut down on so many other things.
yeah,
But
yeah.
of course, you lose out on something else. Yeah.
Well, so and that's a good way to begin. That's a very optimistic note to begin on. And then thank you for bringing that up. But in terms of today, so your forte, your field is structural biology, and I personally feel it's something which is not talked about very much compared to other fields in biology. I that's my personal opinion on it. I think it should be talked about more because structure, as we all know, that's basically structure. It gives structure to everything. And so, if that's proper, then the following lines can be better. And so, why don't we start there? Like, why don't we talk about structural biology? What is it? How would you define it? What does the field entail?
For example, if I take this cup of coffee that I drink, even the way the tip is designed as a function, so that the coffee doesn't spill over. The handle is a function to the cup. In a very similar manner, actually, one can draw parallels to structure and function. The only thing is, what is the function is what changes. So, in the sense that when we talk of biology, your function is the function of the protein
Right,
or function of a machine inside a cell.
right,
So, for example, when we talk about biology, let me take an example of digestive enzymes.
okay,
So, when we all eat food, we eat proteins, we eat carbohydrates, we eat fat. So, all of these has to be digested. Digested. The simplest analogy of digestion is broken down into pieces, into parts, right? So, if you take a long string and if you want to break it down into pieces, you need a scissor. So that's a machine that's actually cutting this long string into pieces. In a very similar manner, the proteins that we eat are basically long strings of amino acids
right,
and a chemical moiety called amino acid. And we are basically chopping this down into simpler units. And that is the function of this enzyme, the digestive enzyme, which we incidentally call trypsin or chymotrypsin, which
right,
are basically what are called a class of proteases. Proteases are machines that cut proteins, and these are called serine proteases under a particular class of protein. But essentially, drawing parallels to it, it's like a scissor that
right,
comes and chops the long string of amino acids into bits and pieces. Now, the way scissor should have a function, like there should be a hinge around which it has to move. Like you have sharpness on the arms of the scissor, and so on and so forth. You have to the machine, the protein machine, the protease, the enzyme has to be designed in such a manner that you need the wherewithal to cut this into pieces. So these are enzymes are basically machines. Every little biological function that has to happen inside your body has to be done by these tiny machines. So we are talking of those machines and the function associated with those machines. Now that function too has to have a specific structure, and without that structure, the function cannot be fulfilled. That is structural biology, associating the structure with the function.
So, when you say a structural biologist, a biologist who's focused on structure, how would he or she, what do they need to expertise in to become become a structural biologist? Is what I'm trying to ask.
So structural biology is a field that has actually began from physics. So it was borrowed from physics.
Okay.
So one of the classical techniques to determine the three-dimensional structures of proteins is X-ray crystallography. And X-ray crystallography has been used by physicists to study materials. And to put it in a very broad term, we are now looking at biological materials. That's the only difference,
All right.
right? So it began with collagen, which is a biological material. It began with DNA, which is a biological material. Then proteins, which are biological materials. Now, crystallography was being used to study, small molecules like salt, like sodium chloride, structures of diamond, structure of various other chemical moieties. Material scientists were using it to look at materials, all sorts of materials, the structure of materials, anything that could be crystallized. or anything that is ordered in nature could be studied using X-ray crystallography. So people used about 70 years ago, people started using crystallography to study proteins, and one of the first of few proteins to be studied is, for example, your myoglobin, the structure of myoglobin that was determined using X-ray crystallography. Now you may ask a question, why do you need such a complicated technique? It is indeed a very complicated technique, where once upon a time, people required to know physics or understand some amount of crystallography and symmetry to really solve structures. But today it has become a black box. It's just like using the mobile phone. I know how to dial you, but I really don't know what is inside that. So that is what this technique has now become. So we've all become users of structural biology, users of.
I don't mean to interject that, but don't you think that's a shame? Like, in terms of
Sorry?
as scientists ourselves, we pride ourselves on our curiosity. At least that's what I'd like to think. We pride ourselves on trying to find the answers which people don't necessarily want to find them out. So when you're using an instrument, isn't it our duty to know what it's about, even though it's not our field?
No, it depends. I mean, see, it's like saying that I'm a cook. I want to cook something in the microwave oven. Doesn't mean I need to know about the microwave oven. So it again depends on what you are. It's a very individualistic thing. So I might be interested in the end product. So as a biologist, I'm interested in what is the structure of that protein that digests the food that I eat, not the technique, the methodology itself. So now that's what I'm trying to say. So either you can view this as a technique, like my microwave oven, or I say that no, this is part of my science and I'm curious about it and I want to learn. The other technique that is used to determine structures of proteins is NMR, nuclear magnetic resonance. The third technique that is coming up in the last 10 to 15 years is cryo electron microscopy. We are compromised on the resolution, but cryo electron microscopy is a very competitive technique that is actually coming up now. Yeah. So that is about the interdisciplinarity. You can see how much of physics is involved in this short 5-minute conversation that I have given you.
Yeah, I mean, I personally love physics because I do not try to restrict myself since I'm a biologist. I stay in my lane because I have had the pleasure of using and continuously been using electron microscope, and having been using that, honestly push myself to understand it because I believe that if I'm using something, I need to understand what it is. And I've seen people do that as well. I've seen people ignore that fact as well. Like you said, who just use the instrument and are okay with it because that's not their lane. And I guess it's a different in perspective. That's all there is to it. But uh, that also brings me to the question of, so we have such wonderful techniques. We have an understanding of why we need to study structure. So it's just one thing is about studying structure and next thing is about manipulating. Once we study something, it's not soon far after do we actually do something to manipulate it that we think is better. So have we actually manipulated any of the macromolecules you mentioned, like enzymes or whatever? Have you ever done that so far?
Oh, definitely. So now once you talk about manipulation, it is to the degree of manipulation that you're talking about. So the smallest degree of manipulation that we have done is created a mutant.
Okay.
And you know very well that we have created many mutants of many proteins. Look at the structure, look at the information that is there in your hand, and then ask which are important amino acids that actually can stop my scissors hinge motion. For example, coming back to your scissor. So if I block this via converting one of the small amino acids that is present at the hinge into a bigger one, my scissor cannot move anymore. So you make the scissor inactive, you see. You've already made a dead mutant. So in a sense, you have engineered, of course, engineered in a manner that this doesn't function. Okay. So if this is a protein that is very essential for a bacteria to function, and I've created a mutant that doesn't allow it to move anymore, I have actually stopped the function of the bacteria. I've successfully stopped the bacteria from functioning. If it's a pathogenic bacteria, then you're closer to winning the war, right? Of making a drug or somehow making it non-functional. So this is the smallest intelligent mutation that you can do because you understand how the structure is related to the function. So the moment you can map structure onto the function, I can go and intelligently, take my screwdriver and fix that one little screw that is necessary to stop the function of this scissor or whatever the function is. So that is basically the advantage of structural biology, and that stems into something called structure-based drug design.
So can you take us through your thought process of, let's say I give you a protein, which, so now you have to figure out its structure and figure out its function. So in your mind, what does it go? Okay, how do I break this down and how can I go forward?
So first thing I would ask is, what kind of a protein is it? So this has to now tie up with a whole lot of bioinformatics, where you have databases of information, all kinds of information about proteins. So I map what kind of a protein is it, and find out that this is a protein that looks like it belongs to the family of proteins that actually cuts amino acids proteins into smaller amino acids. So this is a family of proteases, right? So for example, the coronavirus has a domain which is actually doing the same job as a protease. Okay, so in principle, I can identify that this belongs to the family of proteases. Then I can ask, among the family of proteases, what are the different kinds of structures that they have? Okay, so scissors come in different shapes and different sizes. I can now map and say, okay, this is this kind of a scissor and not that kind of a scissor. It's this kind of a protein and not that kind of a protein. So I can map this, and then I complete, first of all, asking questions using bioinformatics and trying to gather as much information as possible. Once I've done that, then I can map and ask, okay, this is the region that is most important. I can use recombinant DNA technology, overexpress this protein in bacteria or whatever. There are many systems. You can clone, overexpress, make a huge amount of protein, and then go and use X-ray crystallography techniques or NMR techniques or cryo electron microscopy techniques, whatever is amenable at that point of time. Crystallize the protein if that requires to be crystallized, and then go and see if you can determine the structure of the protein. And once you determine the structure of the protein, which is easier said than done. After a few years, let's say you have the structure, then you map this structure onto whatever your original hypothesis is. That is, does this look like this scissor or that scissor or that scissor, or is this a novel scissor?
Right.
All of them are cutting proteins. All of them are proteases. But is there something new about it?
Right.
Then I can borrow ideas that have been implemented for the other scissors, other proteases, and see whether one of those ideas works to inhibit this particular protein. Otherwise, I'll have to develop new strategies. Right. So it's a whole lot of an involved field in itself. You will see that most of the Nobel Prizes that come in chemistry, if you just scan through in the last 20 years, 5 to 6 are in structural biology, goes to structural biology.
Even the latest one is on CRISPR RNA, right? If I'm not mistaken.
Yes. Yeah.
So
anyway, I've answered your question, I suppose.
Wonderfully, in fact, in the sense, like, you not just talked about how structural biology comes in place, you also talked about the research methodology behind that process if you give a problem. And I'm glad he mentioned it takes a few years, because if viewers watch this and be like, it's so simple, you can just finish it. So I'm glad you mentioned that it takes a few years. But I'm also wondering, like, to put it pretty vaguely, can this be used against the coronavirus? Like, is there any opportunity at all? Like, because I personally don't think we can fight this with a vaccine. That's my personal opinion. Because the number of people are
already trying. So people have already come a long way. If you actually scan through literature, early March, April, you would already see papers talking about the structure of the S1 protease of the coronavirus. So that is your first step to understand, how this S1 protein is actually interacting with the AC receptors, and so on and so forth, and to understand the structural makeup of the virus. What kind of proteins are there on the outside? How is it similar or dissimilar to the other SARS virus? All of this is being done.
Like I said, I read a few papers early on about the structure of the virus and how it is. Then I started to see scarcity of those papers as the months went along. There are more papers on how to produce vaccines and stuff like that. The ratio started to differ. That's what I'm trying to
Yeah,
communicate.
because that's more important to make the vaccine now than to get to the structure. I mean, having said that, it's not that there is any shortage of efforts on
Right,
that
right.
side, on that front. People are doing it. There are a huge number of structural studies on coronaviruses, and they've been doing this. But I think today vaccine is more important, so people are trying very hard to look at the vaccine. Also, the market would be very high for a vaccine. There is an economic angle to it, so
Yep.
people are being more for a vaccine, which is what we want.
So, in order to put the conclusion to this interview, I'd just like to ask one final question to you is that, in terms of structural biology, what do you think the scope is for the people who are coming up? Like, let's say they have an interest. So, I would give my own example. When I'm just talk about neuroscience, because that's what I want to do. Sometimes I do focus on the structural part of how the cells are assembled, how they are so plastic. That is a
Mhm.
structure, and also about some of the enzymes which are present in. That's also structural biology. So, what do you think the scope is for anybody who's coming up or wants to be part of this field?
And the scope is wide. I mean, because the complexity of problems that we have started dealing with is also increasing exponentially. So there was a time when we used to look at single proteins. Now we look at macromolecules. There was a time when we used to, macromolecular complexes. 10 years ago, the Nobel Prize was given to Venki Ramakrishnan and others for determining the structure of the ribosome, which is a complex macromolecular machine. Now, in a very similar manner, 10, 20 years ago, people used to make single mutations. Today, we are combining structural biology with synthetic biology to make completely artificial enzymes. Completely artificial enzymes. We are talking about how do we make an enzyme that does a chemical reaction which was otherwise not possible. Okay, so enzymes are catalysts. So, but not every reaction is present in enzymes. Every chemical reaction is not present in enzymes. So, can I actually take a chemical reaction and fix it into the active site of some other protein? This is the field today. People are saying in one active site, can I make two reactions happen simultaneously? So today we are very closely working with synthetic biology, and the growth is exponential.
I think that covers everything. I think we covered everything from what it is to the scope of it and everything that's happened, how you approach a problem more than anything. And that was a well-rounded interview, in my opinion. So I hope you had fun. It was really fun for me to learn it. And my personal view on it is that, you get to use cool stuff, man. Like, electron microscopy, it's fun to use. I'll be honest with you. And so that's my take on it. And so I hope you had fun. I hope it was good for you, sir. It was really
Thank you very much.
fun.
Thank you for this opportunity. And yeah, so we will hopefully have more such discussions and interactions in future. All
For
right.
sure, sir. For sure. Like, I really do hope that. So for this show, thank you for being on this episode. Thank you so much for coming on.
My pleasure.
So there you have it, ladies and gentlemen. Structural biology, its importance, and why you people can actually go and do something about it. Because at the end of the day, it's all about curiosity and what your interests lie in and what you want to do. And structural biology is a very, very cool field to be a part of. So I hope this interview and I hope the information you provided was useful to you, and I really hope you'll do something with it. So go do something with it. But wait. Before you do that, just for courtesy's sake, click on the like button, do share this video, and do subscribe to our wonderful channel, man. Because hey, it's really cool. And in the meantime, stay curious. Thank you so much.